Now, let us discuss about the stability of structure of protein. There are 4 structural levels in protein. First is primary structure. In primary structure, the amino acids are linked in a linear sequence by peptide bond only. The number and sequence of amino acids in polypeptide chain, it is the characteristic of primary structure, and these are linked with a very stable covalent bond, that is a peptide bond. While the another structure is secondary structure. In secondary structure, long polypeptide chain fold or coil to form either alpha helical structure or beta pleated structure. These secondary structures are stabilized, produced and maintained by specially hydrogen bond. First we will see about alpha helix. In this alpha helix, the chain is coiled spirally, generally in the right handed manner, and this helix is stabilized by hydrogen bond between carboxylic acid of one amino acid and NH2 group or amino group of next 4th amino acid. Another secondary structure is beta pleated structure or beta pleated sheet. In this, 2 or more chains are joined together by intermolecular hydrogen bond. So, for secondary structure, hydrogen bond is the main bond which stabilize the secondary structure, and hydrogen bond, it comes under non-covalent bond. So, we can say that in the stabilization of structure of protein, mainly covalent bond and non-covalent bond, they play important role. Peptide bond and disulfide bond or disulfide bridge, can you see here? This disulfide bridge, it is specially formed between the amino acids which has SH group, like cysteine. These cysteine, they join with each other and there is a formation of cysteine, that is SS linkage, disulfide linkage. So, we can say that peptide bond and disulfide bond or disulfide linkage, these are the covalent linkage which stabilize the protein structure, while all other like hydrogen bond which is present between carboxyl group and amino group of another amino acid or between two polar groups, hydrogen bond, van der Waals attraction or electrostatic interaction, hydrophobic interaction, ionic bond, all these are the non-covalent bond. So, these bonds stabilize the primary, secondary, tertiary and quaternary structure of amino acid. See, tertiary structure, as I told you, tertiary structure, it is the coiled structure of coiled and 3-dimensional structure of protein in which the polypeptide chain may undergo coiling and folding to form a ball like tertiary structure. These structures are stabilized by several types of bonds, namely hydrogen bond, ionic bond, van der Waals interaction, covalent bond like disulfide bridges and hydrophobic bond. The fourth type of structure of protein is quaternary structure. Protein is said to be in quaternary structure if they consist of 2 or more polypeptide chains united by the forces other than the covalent bond. The forces which stabilize these quaternary structures are hydrogen bond and electrostatic bond. So, we can say that these are the main bonds which stabilizes the protein structure, like peptide bond, ionic bond, disulfide bond, hydrogen bond, hydrophobic bond. So, we can say that for primary structure, peptide bonds are responsible to stabilize its structure. For secondary structure, hydrogen bonds. In tertiary structure, disulfide bridges, van der Waals interaction and ionic bonds stabilize the structure, while quaternary structure is stabilized mainly by hydrogen bond and electrostatic bond. So, protein structure, whether it is primary, secondary, tertiary and quaternary, they are stabilized by some covalent bond and some non-covalent bond. Now, let us start with the next topic, that is denaturation of protein. Now, what do you mean by denaturation? The phenomenon of disorganization of native protein structure is known as denaturation. We discussed that protein, its structure is quite complicated and it is studied under 4-level organization: primary, secondary, tertiary and quaternary. Each level has its own characteristic, and in that these structures are formed and stabilized by various types of bonds like covalent bonds and some non-covalent bond. If these organized structure of proteins are disorganized by any mean, if these structures are disturbed, then that phenomenon or that process is called as denaturation of protein. See here, this is the diagrammatic representation of denaturation of protein. Here it is a normal coiled 3-dimensional structure of protein. In this, when it undergo denaturation by some means, all the bonds which are present in native structure of protein, which stabilize the protein structure, all these bonds open up and we get a denatured protein, because all coiled structure, beta pleated structure, 3-dimensional structure, they become free from each other as all bonds are break down and we get denatured protein. Denaturation actually it results in the loss of secondary, tertiary and quaternary structure. Primary structure, it remains intact during the denaturation, because in primary structure amino acids are linked with each other through a strong covalent bond, that is a peptide bond, while secondary, tertiary and quaternary structure, they are linked with each other through the weak secondary forces or weak non-covalent bonds. Denaturation result in change in physical, chemical and biological properties of proteins. Generally, there are 2 types of agents which result the denaturation of protein. One is physical agent. Under this, heat, violent shaking, X-rays, UV radiation, they comes under physical agent, while some chemicals like acids, alkali, organic solvent, ethyl alcohol, salts of heavy metals, urea, salicylate, these are the chemical agent which denature the protein. We can take a very good example of denatured protein like egg. When eggs are heated or boiled, you can see that its property get totally changed, because the proteins which are present in egg after heating by physical agent, they undergo denaturation and its physical and chemical properties get changed. What are the characteristics of denaturation? First characteristic is the native helical structure of protein is lost. Because of denaturation, helical structure is lost and it becomes a because all secondary bonds or non-covalent bonds are disturbed, So, helical structure is disturbed. The primary structure with peptide linkage remain intact because peptide bonds are strong bond. So, there is no effect of denaturation on peptide linkage. The protein loses its biological activity after denaturation. Denatured protein become insoluble in the solvent in which it was originally soluble. Before denaturation, if it is soluble in particular solvent, but after denaturation, it won't be soluble in that particular solvent. The viscosity of denatured protein increases while its surface tension decreases. Denaturation is associated with increase in ionizable and sulfahydryl drugs groups. This is due to the loss of hydrogen and disulfide bonds. After denaturation, proteins are easily digestible. Like egg, the example which we have taken, or meat. These are not digestible before heating, but after heating, the proteins which are present in egg, meat, or pulses, they undergo denaturation and these proteins are easily digestible. Denaturation, it is usually irreversible. Once it is denatured, again it cannot bring to its original shape and structure, while denaturation proteins cannot be crystallized. So, these are the characteristics of denaturation. Now, in next lecture we will see about the classification of proteins.